Good morning, everybody. Today's lesson is about 3.1, which is genes. So, let's review. Actually, first. So, the first thing to review is DNA. So, what does DNA look like? Well, deoxyribose nucleic acid. And there's also ribose nucleic acid. I'm going to write messy and sloppy, so you make sure you're listening and watching this over and over again. And I will try to write neatly. So, we have DNA, we have RNA. Please remember, the central dogma of biology is DNA becomes RNA becomes protein. All right? Now, there are 5 nucleotides, right? A, T, C, G, and U. U is for uracil. This is RNA only. Then we have guanine, Guanine, cytosine, Cytosine, thymine, Thymine, and adenine. Okay? So A, T, C, G, and U. Also, as review, so let's kind of do this. We have DNA, we have RNA, all right? So this is ATCG, this is AUCG, this is double-stranded, this is single-stranded, all right? RNA is self-replicating. This is important. Note, just because when we get to evolution, this is one of the reasons why RNA is hypothesized to have evolved first, because it can self-replicate. It's single-stranded. Okay? There's a few types of RNA. We have mRNA, which is for messenger. We have tRNA, which is for transfer. And we have rRNA, which is for ribosome. So messenger, transfer, ribosome. There's 3 types of RNA. We'll get more into how DNA replicates when we're back face to face. So, that's DNA, RNA. Okay, that's kind of like, okay, a nucleotide. So, you don't need to know, it's not like amino acids or sugar, we gotta be able to draw it super detailed. So, we have a 5-carbon sugar. We have a nitrogen base. And then we have a phosphate group. All 5 of these can become triphosphates. So remember that ATP, adenosine triphosphate, is the energy currency of the cell. However, all of the nucleotides can become triphosphates. So there's ATP, GTP. This is the energy currency of the cell. GTP is sometimes used, and TTP, CTP are not used as currency in the body. But they are present because they have to make DNA. All right. Now, as far as 3.1 goes, so the first thing in 3.1 to understand is that what is a gene? So I use this analogy a lot. So if DNA is our alphabet, the gene is a sentence, you could say alleles, which are different forms of a gene, would be like you can say like a different sentence. Like it's like basically, I'm trying to say the same thing. I'm trying to say the sky is black, but instead of saying, like, one gene could be the sky is black, and then the next allele of that gene could be the sky is not white, or sorry, is not is. You're trying to say the same thing. Hold on. You're trying to say the same thing, but you don't say the exact same sentence. So that's what I'm trying to get. Okay. All right. Forget that analogy for now. All right. I messed that up. So, but there's more than one way to say the same thing. That's what I'm trying to say. So that's alleles. Okay. So then we have chromosome. So chromosome would be like the chapters in a book, and then the genome would be the book, right? So every cell has the complete book. Every single cell in your body, every single cell in an organism has the complete genome, right? What makes the difference between a heart cell or a lung cell or a liver cell is the genes that are activated. So different genes are activated. Some genes are only activated when the when the embryo is developing, and so on. Okay? So, what is a gene? A gene is also, like, physically, it is a length of DNA that codes for a protein. Simple as that. Okay? You can also add in some keywords, like a gene is heritable, right? It must be transmitted from parent to parent, or from parent to offspring. So it's a length of DNA that codes for a protein. If it does not do that, then we're not talking about, it's not a gene. All genes code for a protein, okay? All genes have a particular loci, which means location, on a chromosome. If we look at a chromosome of a human, or sorry, I shouldn't say human, just look at a chromosome like this. This is the P arm, this is the Q arm. So, like certain genes, so if you have different genes on here, right, this might be like 3P11, and this location is 3Q12, or 2Q12, whatever. So there's a P arm and a Q arm. Now, difference between, so we have bacteria, and we have eukaryotic. Now, the chromosomes on a bacteria are circular, they're a loop, and eukaryotic, they are linear, right? So you know that humans have like 46 chromosomes and so on, and that blah, 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 blah. So eukaryotic chromosomes are linear. There could be many of them, and there's only one chromosome for a bacteria, all right? These are, this DNA is associated with protein, and this lacks protein. All right? There are single-celled organisms. There's a third class. They're also single-celled. They're called Archaea. They're also like bacteria. But however, you know, their chromosomes actually have proteins. So we'll get into an evolution, but there's 3 different domains. So all life before we get to animals and plants and fungi and protozoa, there are 3 domains of life. There's Archaea, which are also single-celled organisms. There's bacteria, and there's eukaryotic. So, but this is evolution. So we'll get to this. But the important thing to know is that any single-celled organism, that's basically, it's a prokaryote. If it's prokaryote, right? The chromosome is circular, there's one, and it lacks protein. And eukaryotic is linear, many, and associated with protein. All right. Basically, when I'm talking about this, when I'm talking about associated with protein, what happens is, so you have your DNA. So these are called histones. So this is a histone. A histone is a protein. So these are your histone. And what happens is the DNA is wrapped around it and wrapped around it. And these serve two functions. The first function is to pack DNA. It's how we pack DNA into chromosomes and pack DNA tightly. It's like packing a bag before you go on a trip. The another function is it's used to activate genes. So if you imagine it, if your gene is wrapped around a histone, it can't be active because the proteins to copy that DNA into RNA into a protein can't be accessed. It's basically you packed your bag for vacation and you don't want to go into your back. You have to unpack your backpack to get your clothes to change. Same with the histones and genes. If the gene is packed into a histone, around a histone, there's no space for the proteins to get in there to copy that, to read that DNA molecule, make a protein. So many parts of your DNA in your cell that's not active, your cell just wraps around a histone and leaves, and it's like storage. It's like in storage, it's packed away, it's not going to be activated. All right. The next definition is alleles. Now, alleles just mean different forms of a gene. Now, the simplest allele is it present or not present? So, you might have, now, we all have a gene for insulin, right? And there's some genes that we all have. So you might have a form of a gene. You just might not, like, we're talking about type A, type B, right? Like, talk about blood, right? So blood, right? There is A, B, and O, right? I am type A. I don't have this gene. I have A and I have O, or I have AA, right? So that's the simplest version of alleles. But it could also just be, you have like white, black hair, brown hair, blonde hair. So alleles are different forms of a gene. How do we get new alleles? New alleles come from mutation. Okay? Now, there's three kinds of mutation. There's a few. Let's talk about mutations now. So, mutations. There is a point mutation. This is also called a SNP, which is a single nucleotide polymorphism. Okay? Simplest mutation is that the A gets swapped for a T or whatever. That's a point mutation. Okay? There's a few, just like there's many ways to say the same thing. I can call it a point mutation, right? I can call it a single nucleotide. See, these are all alleles of how to define a point mutation. You can say point mutation, you can say single nucleotide polymorphism, or you can call it substitution, because we're substituting one base for another. Okay? Now, the other kind of mutations, with this, there's two types. Okay, sorry. That's it. Then there's frame shift. Now, frame shift are important because your DNA is read in triplets. So here's my DNA sequence. A, T, T, A, T, C, C, G, G, C, C, T, A, T, G, C. Where do you start? Anybody know? What? What? Where do we start? I don't know. Do I start ATT and go TAC, GGC, CTA, TGC? Oh, look, I did it by threes. Perfect. Is that this is called the reading frame. Oh, wait. Could it be TTT and then ACG and then GCC and then TAT? Or, oh my God, is it TTA, CGG, CCT? Where do you start? I have no idea. I just see a big piece of DNA. I have no idea how to start. Well, I'm going to tell you. We all have the exact, all proteins start with methionine. Methionine is an amino acid. Methyinone. I think that's spelled correct. Something like that. Methionine. Okay. The RNA code for methionine is AUG. The DNA code is TAC. Okay, so all proteins start with this sequence. So this is called the reading frame, right? So there's two kinds of frame shift. I can either add a base or the opposite, delete a base. So there's addition and deletion. Now these are bad because, look, simply if I remove this base, every single amino acid down the chain, all this is now going to be screwed up, right? So frame shift mutations, these guys are bad because once you have a frame shift mutation, everything after that gets messed up. So these are bad. Okay, another kind of mutation are chromosomal. And so basically, know this word, non-disjunction. This is basically chromosome mutations, and these are bad as well. Basically, if you have an odd number of chromosomes, it's not going to work. So chromosome mutations. So this non-disjunction can be addition, it can be deletion. It's just any kind of, it's basically when chromosomes do not separate evenly. So we've talked about a few before, like Down syndrome, like Down syndrome. This is called trisomy 21 because you have 3 number 21 chromosomes. Okay? Another example of non-disjunction is Klinefelter's, where I believe this is XXY. So you have XXY. So there's a few of these. You can know a few examples. Okay. Now, 3.1 comes to the Human Genome Project. I love talking about this. I actually worked for the guy that ran the U.S. side. Now, literally, okay, so this was, okay. I know it's really, okay, where's my pen? All right, okay, this is that's not my pen. There's my pen. Okay, God dang it. Okay, all right, this is literally 2000 to 2005. Okay, when I was in high school, when I was your age, I learned there was 100,000 genes. Okay, when I was in college, nope, there's about 22,000 genes. And the human genome, the sequence of the human genome, it took 5 years, and it cost 1 million, or maybe $1 billion. I don't know. It cost a lot of money. It was like in the million. It was millions of money to sequence the human genome. Then, when I started teaching about 10 years ago, that price was about $5,000. So 5,000 USD gets you a human genome sequenced. And I literally heard this on a podcast literally like last week. That price, for $1,000, you can have the human genome sequenced. I don't know the time. I mean, maybe it's, I don't think it's not like immediate. It's not going to be like a day or two. I think it's going to take, I don't know how long it takes, but it's down to $1,000 per human genome. So if you think about it, it's 2021, right? This was 2005. So It's 2021 now. So and what's absolutely amazing, like there's if you go to PubMed, and if you go to get the acronym right. Okay, if you go to PubMed, or if you Google NHGRI, National Human Genome Research Institute, they have a huge database, and you can read about the Human Genome Project. Oh, I'm going to put a link to this. There we go. I'm going to put a link to this in our... Okay, so go to NHGR. It's a genome... Okay, the website is genome.gov. And so, some history. The NIH is the world's largest conglomerate of research. It is U.S. government run. The NIH is made of like 30 different institutes. So it's the largest conglomerate of research. So like, for example, you have like the NCI, which is the National Cancer Institute. And you have the National Human Genome. And what they do is they sequence genes. So they sequence frogs and pigs and cows, and so you can go into his database and you can literally just go, I want to see the insulin gene for cow, pig, human, fruit fly, C. elegans, and you can compare them. And we can do evolutionary studies. We can look at like how conserved is, like when you're breaking down glucose, the genes to break down glucose exist for everything. Right? Everything breaks down glucose. So those genes that are in a worm or a fruit fly or a bacteria are the same genes that break down glucose in you. And so so we can look at, so that's the point of this. And so what we have to know is kind of like impacts. So impacts, right? This is kind of like what we got to know about, because we have to know the Human Genome Project basically it's esoteric, right? It's abstract. So like, what are the impacts of the Human Genome Project? So basically research, this is basic research, right? We're talking about the ability to sequence all genomes. We can do evolutionary studies. We can identify genes. Okay, I'm sorry, being really sloppy. Basic research. We can sequence all genomes. We can compare genomes and genes. So we can do evolutionary studies. We can identify genes. We can find new therapies. Here's the new thing. This is the new thing. Individualized medicine. Because if I'm your doctor, literally you can do counseling. There's a new field of genetic counseling, right? You can go for $1,000, have your genome sequenced, and you can have some doctor and sit there and go like, well, look, you have a high risk of cancer. You have a high risk of glaucoma. You have a high risk of eye problems, blah, 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 blah. So there's this whole new field of like counseling, and I can target medicine individualized, towards you. This is like what is the cool thing about the Human Genome Project. If there's any field of science that is having its golden era, it's biology right now, because where we've gone from 20 years, I was in college in 2002 looking up genes on a database on a website, and that was like brand spanking new in 2002, 2003. And now we're 15 years later, and we have about 5% done. We have about 5% of the human genome identified, figured out, we know exactly what it's doing, blah, blah, blah. So we still know in 2002, it was like 1%. So maybe by the time, 2100, we might have like 50% of the genome done. I don't know, but still a long way to go. But this is the impact of the Human Genome Project. Okay, the last thing we're going to talk about in 3.1. Woo, look at this. This is really long. Look at this. Okay, all the way back to here. We're going to go all the way back to Here, mutations. There is one example of a mutation you must know. You have to know this. I am sorry in advance. You need to memorize this. Where is it? Okay. Sickle cell anemia. Sickle cell anemia. All right. This is a SNP. This is caused by a SNP. So the normal sequence, this is a single base substitution. And basically the normal sequence is CCT GAG GAG. All right? That's the normal. And so this gets transcribed into RNA. And so the RNA this is the DNA. Okay. This is the one you need to know. This is GAG is normal. Okay? Now, what happens is the mutation becomes GT. I'm sorry, I'm going to go over here. Okay, so the normal, we have normal and we have mutant. Now, normal is GAG. The mutant is GTG. Yes, you have to memorize this. I'm sorry. The normal amino acid is GLU, which stands for glutamine. Okay? This mutation leads to valine. This is a different amino acid. Okay? Now, where this occurs in hemoglobin. Hemoglobin is a protein. Hemoglobin is made of so hemoglobin is a protein. It looks like this. A hemoglobin is 4 different proteins together. So we have 2 alpha strands and we have 2 beta strands. So hemoglobin is 4 different proteins. So there's 2 alpha, 2 beta. You put them together and you have hemoglobin. And in the center of hemoglobin, you have iron. And this is hemoglobin. What it does is it carries oxygen in your blood. Okay, I'm back. I was messing you guys. All right, so hemoglobin. Okay, so this is, yes, memorize this. This is the normal. This is the mutant. Okay, sickle cell anemia is a single nucleotide polymorphism. This is the mutation. GAG becomes GTG. The amino acid is glutamine. The mutant amino acid is valine. And what this does to so hemoglobin is a protein. It carries oxygen. Now, the reason why I mention all this is because for HL, basically, because HL needs to know this, because this is quaternary structure, right? In a protein, this is HL real quick. HL. In a protein, the primary is the sequence of, actually, this is good for everybody though. So just listen. So primary structure of a protein is a sequence. So this is glutamine, valine, methionine, whatever, right? Sorry, methionine is always first. So alanine, right? So that's the primary sequence of a protein, is the sequence of amino acids. The secondary structure of a protein is going to be a helix, a spiral, or it's going to be pleated, like this is pleated. So it's like a wave, right? That's secondary structure. Tertiary structure of a protein is the shape it takes. So, for example, this is globular, right? So globular would be like a circle, a globe, right? Proteins could be shaped like a spiral or whatever, but the tertiary structure of a protein is how the protein shapes. The quaternary structure of a protein is this, where you have 4 different 4 different strands. You have 4 different wires, and then they're bonded together. So there's bonds. There are different bonds that hold this all together, right? So this is quaternary structure of a protein. I bring this up for HL because you have to know this, but for ASL, it helps you to visualize what a protein is. So what does this single mutation do? The single mutation, what it does is it makes your red blood cells look like this. Okay, so this is normal, and this is sickle cell. Now, why did this persist? Why do we still have this around? This is bad. Sickle cell is bad. You don't live very well. You die young. You have problems. Why do people have sickle cell anemia? Well, then comes along Plasmodium. Now, so we have malaria. Now, malaria is caused by a parasite, and the parasite is called Plasmodium. Okay, so Plasmodium is a parasite. Plasmodium can attack normal cells, but it cannot attack sickle cell anemia, right? And Plasmodium carries malaria. So if you live in an area, so basically here's this. So this goes back to if you are, so HBS is the sickle allele, and HBA is the normal allele. So if you are AA, you are normal. And if you are AS, if you're a heterozygote, you are a carrier. And if you are SS, you have sickle cell anemia. Now, so this is bad. This is not so bad. And this is normal. So, if you live in an area where there is malaria, right? If you live in an area where there's malaria, the best option is this. Because if malaria is present, you have, if you are heterozygote and you have both sickle cell anemia, not sickle cell anemia, but if you have sickle cells and normals, if you have both of these cells, right, and there's malaria, you have an advantage because you have normal blood cells, right, and you have sickle cells. So you have a natural resistance against malaria because malaria, you can still carry oxygen. So malaria can, if you are normal, right, malaria bad, right? And if you are sickle cell, you have sickle cell anemia. Anemia means lack O2. That's what it means. If you're anemic, you're lacking oxygen. So if you have sickle cells, if you have all sickle cells, right? If you are 100% sickle cell, you can't get malaria, but you have sickle cell anemia. And if you're 100% normal, you can get malaria, but you don't have sickle cell anemia. However, if you're a heterozygote, you don't have sickle cell anemia, and you're resistant to malaria. So in areas in Africa or where there's a high mosquitoes and high malaria, you will find more people are heterozygote. So that's why you learn about sickle cell anemia. So you need to know, so this connects directly to our last topic, which is genetics, right? Heterozygotes. So you should describe, okay, can you draw the Punnett square? A-S, right? A-S, A-S, right? So if both parents are heterozygous, you have if both parents are heterozygote, you have a 50% chance to be a carrier. You have 25% chance to be normal, and you have a 25% chance to be sickle cell anemic, right? Can someone, so if one parent is normal, right, and the other parent is sickle cell anemia, right? Can the child have sickle cell anemia? No, they cannot, right? Because this would all be AS, 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 AS. Now, this is important to know. If you're a heterozygote, if you're AS, you are still normal. You do not have sickle cell anemia. You are a carrier. You can transmit the sickle cell anemia. Sickle cell anemia is the disease. You only have the disease if you are homozygote for both sickle cell traits, right? So 2/3 of the people, so if both parents are heterozygote, that means 2/3 are going to be normal. Their phenotype, their physical traits, they are normal, right? But if you look at the blood, if you're AS, if you're heterozygote, if you look at the blood, you have both phenotypes. You have sickle cells and normal cells, but you don't have the disease. So you only have sickle cell anemia if you're homozygote recessive, which is SS. All right. This just about covers, let me make sure. Okay. The next thing I'm going to do, after this, the last thing, now this is great this is IA talk. IA talk, yay! Okay, in the syllabus, it says you need to use a database to compare sequences. They can't ask you to do that on a test. However, they might give you different sequences. So if I gave you, I don't want that pen. A, T, T, A, T, G, 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 C. Okay, so this is the insulin gene for a mouse. And then, all right, so now here's our sequence. A, T, 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 G, G, C. This is the insulin gene of a human. And then A, T, 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 or A, T, C, C, T, right? This is the insulin of a fruit fly, right? So insulin mouse, insulin human, insulin fruit fly. Now, look, these are all the same. Now, between the mouse and the human, if you look at the mouse sequence and the human sequence, there's only one difference, right? But if you look at the difference between if you compare mouse and fruit fly, there are 3 differences, right? And if I compare, so this is what I'm talking about, the Human Genome Project, right? This is why it's important. So now I can compare the sequence of a mouse, a human, and a fruit fly. And I can look, the mouse and the human are more similar. There's only one difference between mouse and human. But fruit fly and mouse have 3 differences. Or human and fruit fly, they also only have, could have done a little better with that. But insulin and fruit fly might have 2 differences or 4 differences, right? Now, there is a software. If you Google BLAST, BLAST is actually an acronym. If you Google BLAST and make it all capital letters, it's called the Basic Local Alignment Search Tool. So I can give you an IA. I can give you a project, and maybe we do this today if you have time. You can go online. You can go to BLAST. And there's nucleotide BLAST. There's protein BLAST, and I can just, when you say BLAST, basically we're doing a search. So I can put in a sequence of DNA, I can blast it, and I can find, this sequence is in humans, it's in mice, it's in cows, and blah, 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 blah. Okay? On my website, for sure, if you go to DNA Genetics on my website, and you scroll down all the way to the bottom, there is a lab called Alignment Lab, and you can download this software called ClustalX, and you can sit there and line up sequences and figure out how similar they are. There's actually Alignment Lab, and there's also a BLAST Lab, which does the mouse glucose sequence gene. So that's 3.1. Wow, look at that, 3.1. So basically, 3.1 is, what is the gene? What's an allele? What is the DNA structure? This is all review. This is review, okay? So gene, what's a gene? What's an allele? What's a chromosome? Chromosome structure, that's also kind of review. That's connected. basically 3.1, what you need to know is you need to know what genes alleles are, you need to know the Human Genome Project, and you need to know sickle cell anemia. That is 3.1 right there. The chromosome structure, the DNA structure, those are all related to 3.1. And so they might ask you a question where they say, like, part one of the question will be, what is the DNA structure? And then part two of the question will be about sickle cell anemia. And then part three of the question could be like blast or like, you know, they could ideally give you, like, they can't ask you to use a database on a test, but they could totally ask you a question like this where they give you different sequences and ask you to compare them and go, oh, look, the human in the mouse is more similar than the fruit fly in the mouse, and so on, right? And this also makes great for like IA stuff and practical lab skills. I do apologize. You have to memorize this Okay, just memorize it. I had my website open while I was talking to you guys because I don't have it memorized. You have to memorize it, not me. And then the Human Genome Project. This is esoteric. They might just ask you, this might be a few point question like, what is the impact of the Human Genome Project, or why do we care about it, you know? And yeah, non-disjunction chromosome mutations, those are related to topic 3.5, or sorry, this is a related topic. It's not actually 3.1, it's just related to it, 3.1. And we'll talk more about DNA replication and methionine and that stuff later on. Okay.